Okay, so before we actually go into the lecture, I have to give some disclaimers about biology. So, when people think about biology, they usually think about memorizing, but the fact is, well, that is half true because you need to memorize a lot of stuffs and terms, and but what's more important is like understanding what's actually going on. So if you try to like imagine a process, like storyline, how it's going to carry on, it will be much easier to memorize. And the second thing that you is really important is the exact phrasing. So in my lecture, for the ease of your understanding, I'm not going. Of most of the times, I will be using the exact and correct uh, explanations. So please do keep that in mind. And for the exact phrases and words that you have to use, please check the notes that the school has distributed. Yes, okay, so oh, and for this lecture, I'm going to give small recaps of the previous term materials. For example, currently I'm going through term 3 material, and if term 1 material is involved, I will be including them in like necessary parts within this video or and the preceding ones as well. Okay, before we go into the main lecture, I first have to go through cell structure. Okay, for the cell structure, right? So if we say that this is a cell, so we can separate this into three parts. Firstly, it's a nucleus. The second one, which is cytoplasm, and the others are called membrane-bound organelles. But in this case, we're just going to focus on nucleus and cytoplasm. Okay, so nucleus is literally the one at the center. It contains DNA, in other words, genetic material. Okay, for cytoplasm, you can think of cytoplasm as literally everything. So basically, cytoplasm consists of cytosol, so which is like a gel-like watery thing that is inside the cell. And if you remember from term one, there is this cell organelles such as ribosomes, rough endoplasmic reticulum, Golgi apparatus, and all those kind of things. So in cytoplasm, it contains cytosol and all the cell organelles that we're going to take a look at. Okay, so another thing that I have to go through prior to the main lecture is DNA structure. Okay, so how DNA is organized and stuffs. Basically, this is actually taught in term one, but I'll just briefly recap, like really the simplest parts. Number one, if you remember that it's like the simplest form of DNA, well, if other than nucleotide, okay, I'm talking about polymer type, is DNA strand. So DNA strand is simply a double helix structure, so like the latter one you see in cell labs. So this is this is the simplest kind you can ever see within the cell, and this will coil around the histone octamer, all and coil around, condense and everything. Then we get this thing called chromatin. Remember, the behind is tin. Okay, this is the important part. So when this condenses even further, like coil around, like loop around and stuffs, yeah, then we get this thing called chromosome. So this is the most important part. So the difference is DNA strand, chromatin, and chromosome. Okay, so the thing is, the more right we go, the more condensed it is. The more left it is, the less condensed it is. Okay, during the cell phase, right? Um, most of the time, the DNA will be in this state, which is chromatin. So when we draw chromatin, we draw something like this, actually, after replication. So if you take a look at the nucleus, remember we just see one whole color of things, right? It's because it's really thin and spread around, and there's a lot of them. So it kind of like fills the entire place. That's why it looks like a color. Uh, in contrast, chromosome, we can actually see them like like that. So if you take a look at the video of mitosis, you can remember there were some structures, right? Like rod-like things. Those are chromosomes. And the DNA will be in the state of chromosome only during mitosis. And by now you'll be wondering why, then when will we in DNA strand state, right? So it will only be during replication. like partially. So if chromatin is like this, then this part will be like in the DNA strand state, and then the rest will be condensed back again. So as it is being progressively replicated, uh, it's going to open then close up. Yeah, so this is how DNA will be organized within the cell. Okay, so here's another material that you have to take note. Okay, so these are chromosomes. Well, pretty much obvious. Okay, so I'm going to explain some terms that is I'm going to use here. Okay, so this thing I have highlighted blue is called chromatid. Remember it just now was tin and so, and now it's tid. Okay, so this green one, sorry, my mistake. The blue one is going to be a sister chromatid of the one that is going to be the one I have just labeled in green. So these two are sister chromatids, and the one I'm currently labeling it yellow is going to be non-sister chromatids. Okay, and as a whole, these two, we're going to call them a pair of homologous chromosome. So these are the terms that you have to take note. Oh, sorry, there was one more. Okay, so this part I am currently like drawing now. It's called the centromere. So this is the part where it connects the two chromatids, and which later separates in which yeah. And another thing that is on the centromere is this protein. Remember, it's a protein, okay? Protein called kinetochore. So so kinetochore is attached onto the centromere, and centromere is actually it's actually also a DNA sequence, just that usually doesn't code for any functional proteins at all. Now I'm going to clear one misconception that people usually have. So when we take a look at this, people consider this as two chromosomes. Okay, the fact is no, this is not two chromosomes. This is a single chromosome with two chromatids. Remember, you see 1, 2, but they're still connected by the centromere. In other words, this is still going to be called one chromosome, and it's only two chromosomes when counting like this. That's what we call it, a pair of homologous chromosomes. Yes. Okay, so now I'm going to explain about the cell phase. Okay, for cell phase, we can separate this into two major parts. One is interphase, and the second one is M phase. And for the interphase, we can separate this into three smaller parts, which is G1, S phase, and G2 phase. And for the case of M phase, remember it's not a PMAT and PMAT yet. We are going to take a look at mitosis and cytokinesis first. Okay, so these are the two things that separates the M phase. Okay, so try to recall what I taught you about the cell structure just now. For example, just now there was nucleus and the cytoplasm. Okay, remember these two are the things we're going to take a look. Okay, so during interphase, the G1 phase is the part for the cytoplasm. So this is where the cell mass grows. So the cytoplasm doubles in amount. In other words, the cell organelles like rough endoplasmic reticulum, mitochondria, and those kind of things, like including proteins and well, pretty much everything, is going to be doubled. So this is the part for cytoplasm growth. Okay, so in a way, for S phase, this is the part for DNA replication. So this is the part where the genetic material increases. Well, technically it duplicates, so it doubles. G2 phase is the part where it prepares for the M phase. In other words, it starts to prepare. It prepares the enzymes that is required for the mitosis and cytokinesis. And I hope you recall that all the reaction that occur within the body is catalyzed by enzymes, and enzymes are usually proteins. So in other words, we are making proteins that is for the M phase. Okay, so we're going to take a look at M phase. M phase is going to be well, this is the main part, mitosis, and mitosis is separated into four parts, which is prophase, metaphase, anaphase, and telophase. I'm pretty sure you all know that. There's some people also put in prometaphase, but we're going to ignore this for this meanwhile because it just complicates the whole entire story.